Hi, this is Sean from Eve's Temptations Royal Pythons and Eve's Temptations Creations. I chose to sponsor That Wolfie Royals podcast because I believe the hobby needs things like this to bring us together and to showcase all the talent in the hobby, old and new. I am a hobbyist Royal Python breeder specialising in double and multi-recessive projects. Check out Facebook and Instagram to keep up to date, Morph Market for available animals, and feel free to message or catch me at a show for a chat. In early 2021, I set up Eve's Temptations Creations initially to print my own snake bags, and it grew massively from there. I print t-shirts, hoodies, mugs, key rings, glass, photo boards, and much more. This is merch printing for the breeders, by the breeders. Unlike the big companies, I will only print it if it will look good, so I can check your logo files and can alter for free, so there's no nasty surprises when your stuff arrives. I can also do gifts for the family, like photo slates, mugs, and baby grows for all occasions like Mother's Day, Father's Day, and anniversaries. Podcast listeners can ask for the podcast bundle, a t-shirt, mug, and keyring package at a discount, which can be for your own logo, podcast logo, or a mix of both. Find both pages on Facebook and Instagram by searching for Eve's Temptations Royal Pythons and Eve's Temptations Creations, and keep listening to the podcast for limited special offers throughout the year. Thanks for listening.
Hi, it's Dan from World of Exotics. We're a specialist reptile shop based on the Wirral, proud to be sponsoring the Wolfie's Royals podcast. If you're ever in the area and need any of your reptile supplies, we're always fully stocked with live food, frozen food, and everything else you need for your animals. We always try to have a wide selection of different species in store with a good selection of royal pythons. We update our animal lists weekly on our website at worldofexotics.co.uk, and please check out our social media on both Facebook and Instagram at World of Exotics. Now back to the podcast.
Hey guys, what is going on, and welcome back to another episode of the podcast. We are joined today by Dr Ben Morrill of Red Genetics Inc. So all the questions that you want to know about your shed testing, everything like that, he is the man to speak to and has got all the answers, hopefully. I won't talk too much, we'll get straight into this one. So without further ado, let's bring in Ben. Hey Ben, how's it going?
Good, how about you, Tom?
Yeah, I'm not too bad, man, thank you. So yeah, as I said in the intro, you're the guy to go to with shed testing questions, history, background of it all.
Yes. It's been something I've been thinking about for many years now.
So let's get into it then. So where did Red Genetics Inc. come from? Was it, what's your background? Where does your involvement with it begin?
Yeah, so I grew up probably like a lot of people listening to this, catching snakes and lizards and frogs, all that kind of stuff. When I was moving out of my parents' house, I found my journal from 4th grade where my teacher, she had us write 5 lines every single day. And I was writing about catching snakes back then, garter snakes specifically. I'd write down how many I got that day, stuff like that. And so it's just always been there. As a high school student, I called the local university and just cold called. And the biology department said, "Do you have a herpetologist I could talk to?" And they gave me a number and a name. I called, and he was patient and nice enough to deal with a high schooler. With, I'm sure I had all kinds of crazy energy, and but he let me come in and meet grad students and talk, and he could tell like I had done a lot of reading and studying, and then as a undergrad, I got to do some research with him. His name is Dr. Joe Mendelson, and he's the head of the herp department at the Atlanta Zoo now. But yeah, so that's kind of where I got to. Then I went to grad school and I did a lot of molecular biology, mostly for food animals. So cattle and pigs and mice. We did some studies on mice as well to apply it to food animals. But the whole time I was always thinking about how I could potentially use this for the reptile industry. And when I got my PhD in 2011, a good friend of mine that had been online friends for 5 or 10 years, Sean Christian, and he's a serial entrepreneur since he was like 10, 12 years old. He's like, "Why can I send a feather or a blood spot in from my bird and they can tell me what sex it is, but we don't have anything like that for reptiles?" And I said, well, it's because no one's figured it out. And so since 2011, we've been thinking about it. 2017 is when we made the first test available. That was a sex determination test in colubrids. And then 2 years ago is when we were finally able to do both morphs and sex determination in ball pythons.
And where does the data for that come from? So from what I think I've heard in the past is that some of the tests like pied, albino, etc., existed because they were in other animal species already. So the data was kind of transferable almost.
Yeah. So pied was the first one that a group in Canada worked on that one. They took a gene that they hypothesized from what it did in other like mice and zebrafish or other animals where there's been a lot of genetic work done. And they were able to, I think even in that case, they did kind of just this whole genome search.
Okay.
And found that gene that made sense. With albino, that was a group at Eastern Michigan University, Dr. Hannah Siddell and her students. She had a bunch of students that helped her. But yeah, with albino and ultramel and lavender albino, they went to genes specifically that they had picked out. Same with the yellow belly complex. That was a gene that she had specifically picked out that in other vertebrates had shown to do similar kinds of pattern and color changes. So that helped with a lot of the tests. Clown was the first one that we found, and that was another one of those. We just looked at the whole genome of like 60 clowns, and 60 we knew didn't have clown at all, and we used software to pull out any mutations in the clown group that was absent in the non-clown group. And that gave us, 5, 7, whatever potential hits that we then designed tests for and figured out which one was the right one. And then we got a clown test. So there's kind of different ways to come at it, and both have worked over time.
So for the non-science people that are going to be watching and listening to this, what is the process of determining the test? How, what data are you looking for? How are you extracting that data? And how long does it take to make one of these tests or, to find a test that works? Because there must be sort of pitfalls along the way where you try something and it's a failure, and then eventually hit the jackpot.
Yeah. So like what I was just talking about with clown, we had a whole bunch of people send in clown sheds and a whole bunch of people send in, desert ghost and I don't know, hypo, all kinds of stuff. But we just took all the ones that we knew were clowns and any of them that we knew didn't have clown in them at all, and sequenced all of those and then had the software pick out. So when we get that 5 to 7 possible locations, those are 5 to 7 possible actual mutations, like a T to an A or whatever. So we had like 5 to 7 of those, and we designed a PCR test, which most genetic tests have some kind of polymerase chain reaction or PCR that's part of it. And what it really does is it sets a boundary. So like if the mutation we want is right here, we put a primer on each side of it, and then we can zoom in and focus on that one little part of the DNA, and we can see. And then what we'll do is I'll get another set of 50 clowns, 50 het clowns, and 50 that I know aren't clown at all. And I'll test those 5 tests that I designed, and we'll see, okay, test number 1, it called 50% of them right, 50% were wrong. So that's garbage. Test number 2, 70% of them are right, but 30% were wrong. Oh, test number 3, it called all of them right. Or maybe it's one that it called wrong, and I call up that person that sent the shed in, like, hey, could this possibly be, not clown, or could it be a shed mix up? And they send me another shed, and then I do it again. Oh, it is a clown. And, there's definitely different back and forth things that we have to do. But once we get to where we know we've run over 100 and it's, 99% or 100% correct, we always try to make the test available once we know we've done over 100 and it's been over 99% correct. So that's what we're looking for. And there's definitely lots, like with Monsoon, we've gone through, I think we're on the 4th time through sequencing more samples that came in, adding that to the data set, having the software give us hits, then we chase some stuff. And so far it keeps being garbage. And I know, other groups are working on it as well. We're actually, for Monsoon, we're all working together now. We're sharing data because it's been campaign. And it's just kind of this, iterative thing where we just have to keep doing it, get all the hits we can, try them all. If it's garbage, then we have to go back and sequence more and start all over again. And then with clown, the first time we did it, we found, 5 or 6 hits, and one of them was perfect, and it's worked ever since. So it's like I tell people a lot of times, every new morph test is its own journey. Sometimes it's really fast and easy. Other times it seems like an epic journey.
Is it much easier to develop the test when they're allelic combos then? So you're looking at, like, say, like chocolate, spot nose and things. So a rough parameter of where that hit's going to be.
Yeah, yes. Because I mean, all you have to do is find one of them, and then the others are close by. So
Okay.
So that helps a lot. You don't have to be looking in 1.5 billion base pairs anymore. It's likely in that gene or a gene right close by it. So you're looking in, 20,000 base pairs instead of 1.5 billion.
Yeah, that's quite a big difference.
Yes.
So what's like the time frame roughly to develop one of these tests to bring it to market as such?
It depends on how quickly people send sheds in. Like if we go from ground zero, we got to wait till we get enough sheds to come in. But say we're at the point where we have enough sheds here to send them off for the kind of deep sequencing, like whole genome sequencing. We don't have a machine that can do that ourselves yet. That's a $500,000 to $2 million machine, depending on how high throughput you want. So we have to send that out, and that usually takes about a month and a half to 2 months for us to get the data back. Then we have to analyze that, and then we have to take any hits and go after them. So if it works the first time through, 3 to 4 or 5 months would be pretty fast. If it doesn't work, like Monsoon, I think we've been trying for over a year.
Oh, wow. Okay.
Yeah.
Yeah, that's one that a lot of people are going to be waiting on then.
Yeah, and we're all working together on it. People in Canada and Europe, and here in the United States. I think there's a guy now in South America working on it too. There's several of us working together and communicating so that we can figure this out. And it's just a tough one.
And I'm probably guessing here that I may be wrong, and I could be right. I'm guessing there's not really much funding for something like this in terms of genetics or science-based research, because it's quite a niche market.
Yeah, it's 100%. At the beginning, it was Sean putting money up, his own money, and now we do our best. I would say now the majority of the time we are not in the red. We're able to use the sales, so it's really all of you that use our testing that are paying to develop the next test. Thankfully, we're not having to borrow much anymore. We're mostly able to stay afloat. We're definitely not making money because everything that we're bringing in, we're immediately using it to design more tests. I have tried a couple of times to do some applications for grant money and stuff, and there might at some point be some help. It's unfortunate because you kind of have to show your success in that area and how it's helpful, and then they'll like, oh yeah, that is worthwhile, but then you already did the hardest part. But anyway, there might be something eventually for the people in academics. They're able to get some help, like where they're doing their research. That facility is already paid for. A lot of the money would have to go towards building or renting a facility and equipment, and so being in academics helps where some of that's already covered.
Yeah, I can imagine, because they've got, like you say, they've got the facilities and the funding for the base part of it already. What does it roughly cost to create a test?
Since it varies so much, it can be anywhere from 5,000 to 10 times that.
Oh wow! Okay.
We're usually trying to run customer samples alongside the research work. We can't always do that. So some of the time the run is paid for, at least in part, by samples we're running. We have a test that a customer wants results from, and we're able to put that on that run and help kind of cover that cost. So we're not have to always paying the whole cost for research. Since we really started out with that colubrid test, back in 2017, and we just use that money to help fund the next one, and then use that money to help fund the next one. And so it's, if I just sat back and thought how much we've spent on Monsoon and we still don't have a test, it's a lot of money.
Yeah. And then the ones that you do hit, fairly quickly, like the clown, which is obviously going to be huge in the market, and a lot of people are testing for that straight away. It's sort of levels out. But
yeah, so probably the majority of the money that we spent on clown, or that we made back on clown, got spent on Monsoon, and we didn't get anything.
How far away from a test for Monsoon do you think you are?
It could be as soon as the next couple of weeks. It could be, that what I'm trying and the couple other people I know that are working on stuff, like over the last couple of days, we were talking about a couple of sites If that works, we could have it in the next 2 weeks. If it doesn't, we might have to go back to sequencing, and then it's another month and a half till we get that back, and then we've got to analyze it, and all that stuff all over again. I think we're on the 4th cycle now, or either we're on the 4th or the 3rd, one or the other. So anyway, yeah, it could be that we'll have it in a couple weeks, but it also could be, we got to start all over again.
And then the challenge of determining another sequence for testing and stuff starts all over again for another gene.
Yeah. So people ask these questions a lot, and it's unfortunately it just varies a gigantic amount. Sometimes it works the first try, it's really easy, and other times, like the green tree python sex determination test, that's the one we probably spent the most money on that we still don't have. And recently, I think it was last Tinley, we talked to some researchers that do academic researchers that do work on sex chromosomes, and specifically in reptiles. And they said there's something about the genus Morellia where their sex chromosomes are different from other members of the python group. And It would make it so it could be impossible to design a genetic test, or at least very difficult. So at least we have a reason why we've failed at that so many times, but you just never know. It's doing something that no one else has ever done before. And so, if it's like other things that other people have done, then, it might be easy. But if it ends up being a special case, it might be a huge pain in the butt.
Yeah, I mean, that's going to be another huge test as well, because it saves people like 2 years of like raising neonates and struggling through clutch after clutch after clutch of those. And
Yep. Yeah, and Sean and I were both carpet python breeders for many years, did lots of carpet. I actually helped write one of the chapters of the complete carpet python book. And so that was the number one test we wanted, was a green tree python sex determination test in 2011. That was what we talked about. And I tried really hard to get it, and I kept failing. And that's why we did the colubrid one, because colubrid sex chromosomes are very different. So it's a lot easier to find a test that can tell the sex chromosomes apart. And we just couldn't get it to work for green trees. And then, we got at least something going, and we kept going back to the green tree and trying again as we learned more things, and just always fail.
And is it still something you're working on, or have you put that on the back burner a little bit?
It's on the back burner. I hope there will be some publications that maybe I'll be able to learn something from, and maybe be able to. But from what they were saying, it is a possibility that it's just not going to work as a genetic sex determination test. And we already know that for some species that have temperature dependent sex determination, since it's not something in the DNA that causes, like with crocodiles or something like that, that temperature is what affects whether they're male or female. It's not genetic. So we already know for some reptiles we can't design a DNA based sex determination test, but for everything we've ever known with pythons and any pythons, including Morellia genus, it should work. And it might still, but it sounds like there is something unique about the Morellia sex chromosomes that's different from the other pythons. So pain in the butt. Yeah,
that's a headache that I don't envy, that's for sure.
Yeah.
So going back to 2019, when the sort of the ball python market and the test for the ball python market come out, are you seeing a huge number now in people that are sort of an increased number in people testing as opposed to back then?
Yeah, there's definitely a steady growth. It's been really good. There's other people doing testing as well. At first we're really the only ones openly marketing to do testing. Now there's multiple places that do it throughout the world. It's not just in the United States. There's Europe and Canada, at least, that I know of. So yeah, it's definitely been a steady growth. Plus, we keep getting more and more tests that are available. So, things that people are testing for today that wasn't even an option a year ago because there was no test for that. So I think that very quickly within the next month or two, we should be up to like 50 ball python morph tests. I think we're at like 35 right now, and we've got quite a few more coming pretty quick. We'll be between 50 and 60 ball python morph tests, like I said, in the next month or two.
Oh wow. Okay, so yeah, that's quite a big jump then compared to what's there already.
Yes, yeah, lots coming. Just next week we'll be announcing a pretty healthy list.
Okay, I'll look forward to that. That'll be interesting to see for sure. So how do you think that the testing is sort of changing the way that people breed and keep the animals now?
It's been fun to watch because some of the things, how it would affect people, I kind of predicted, like, because I've been breeding. I hatched out my first ball pythons in 2003, and I've had at least one clutch of ball pythons every year since 2003. So this is my 21st year hatching out ball pythons. So some of those were predictable, but some of them, people are using them in ways I hadn't even thought of, and so it's been pretty cool to see. But I think I think probably the most beneficial thing that I think some people didn't realize at first is that it really, people thought, the test, oh, now you're going to test, there's going to be so many babies that are going to get culled or sold cheap or, whatever, because of all these posets. But really, when you think about it, that's like at the point where you're testing adults. But when you're able to test babies, then you don't even hold back or grow up the ones that don't have the genetics you want. So, now that we're a couple years in, people are able to have a higher percentage of their clutches be the genetics that they wanted and they thought they were going to be, instead of, posset to posset and stuff like that. And where you have either the whole clutch or a majority of the clutch is unwanted genetics, now you're able to really slim down and focus and, be able to get specifically the genetics you were planning on. And so it also makes it so you can get a lot more incomplete dominant traits into a recessive or double recessive or triple recessive project, because you can get a male that's a het or double het or triple het and breed them, and then you can just test for those recessive genes in the baby. So, it makes it much easier to figure out what holdbacks you'll have. One of the aspects that I didn't expect, that Justin Kabelka, he said that it's made it so he can sell babies faster, whether they're het or not. He said it's been really nice because he's found that if he puts an animal up, it's a combo, it's, posset clown, posset pied. But if the people go in to buy it, they're like, well, I only work with clown, I don't need pied at all. And, I'm probably in their mind, they're thinking I'm paying this extra amount because it's posset pied. And so they're a little less likely to buy that animal just because it's a posset pied. So he said that if it ends up, proving it is het for both, he can post it as that and people know it has both. Or if it's negative for both, he can post it for that and people know what it is and they know what they're getting. They don't feel like they're wasting some money on a posset that they're not even going to use in their collection. And so he said it's helped be able to sell animals quicker because, people know they have a higher, probability of knowing exactly what they're going to get, and meeting that expectation is really good for both the seller and the buyer.
I know Justin's doing this quite a lot as well, and he's putting a huge amount of samples in for shed testing. Are you seeing other people put in large chunks and huge quantities at the time, or is it still just more individual animals that they're looking for?
Well, there's definitely many people will get 30, 40, 50, 80 sheds at a time, multiple times through the breeding season. There's definitely many people that are they tell us all the time, I have changed my my breeding plans since this test came out or that test came out. They'll contact us and be like, hey, I know monsoon's coming soon. Is it going to come in the next 2 weeks? Because if it is, I'm going to hold off putting this male. There's all these things that it can affect when you're really trying to, push and get the most cutting edge genetics that you can in your offspring. So yeah, there's many people that are doing a lot of testing and utilizing it to really get a lot of these incomplete dominant morphs into these double, triple, and quad types of projects.
Well, that's the way everything's going at the moment, isn't it? It's like the triples and the quads. So like you say, with you can buy a triple het male, which is still relatively low priced, and you've only got to pair that to anything and you can test it. So
yep,
it definitely adds value to those animals that were perhaps sort of as maybe lower end or undesirable at the time.
Yep. And then the ones, sometimes you'll get 7 females in a clutch. You're like, dang, they're all pos triple hets. I don't know which one to keep. But now you can test, and some of them may be you just give them to the pet store, with a couple others that they buy, or give it to someone as a pet or whatever, because it's not het for anything, or it might just be het for one thing or whatever. But just 2 or 3 years ago, you would have kept all 7 and, at least waited till you bred a few and proved one of them out before you sold any of the others, because you don't want to miss out on that triple het girl,
Yeah, that makes sense. One thing that we had before when people would say, you got het pied markers, or, this is what they're looking for in het clown. Are you seeing, obviously, you're not seeing the people's animals that they're sending in, but are you seeing that people are saying, oh, these has got strong markers for something, testing, and it's not coming back as that, or?
I've seen both. With pied specifically, I've seen people send in and they'll write in the notes. They're like, this is not from, a pairing where the pied is in there, but it's got such a big marker. And I've seen it go both ways. Sometimes it'll be, yeah, it is a het pied. And then sometimes it'll be, nope, it's not. And there's one specifically, I think it was a sunset, or it was something, it was really cool. And it had this, just beautiful, probably 1/4 inch to 1/2 inch thick, marker. What do we call that? Ringer. Ringer. It was a thick ringer. It looked stereotypical, as much, het pied marker as you could hope for. But it wasn't from a pied breeding. Like, yeah, you might as well test it. That would be awesome. And it ended up being negative. It just had that ringer for whatever reason. So it can go both ways. If it's a decently valuable animal, like if I hatched one out I didn't expect to be het pied, and it's got, 2 or 3 other things going on, I would test it just in case it's floating,
Yeah. There's so many animals as well that have been bred over the years with unknown hets in them, so who knows what's going to pop up. Albino pops up in a lot of axanthic stuff because people were chasing the snows years and years ago, so that bloodline's still going to be in there at some point, and you might just get unlucky and hit the 2 the 2 and 1 that you perhaps didn't want.
We've had het clown, het pied, het candy, het albino. We've all had all those pop up where people didn't expect them, and sometimes they'll go back to the breeder. I remember 2 in particular were 2 different people, and they said, oh, that came from Bob Vu, and they went back to him. He's like, oh yeah, I do have that down as Pos het clown. I just never proved him out, and so I didn't say anything, but yeah, it makes sense. So not only did we prove that baby to be a het clown that the customer wasn't even expecting, now they're so happy with Bob Vu, they got that extra gene, and now Bob Vu knows that male is a het clown.
It's a het clown, yeah, of course. I didn't even think about that knock-on effect of going back to the breeder and chasing up the parents and things.
And seeing your shirt reminds me, Clutch has done an awesome job with that. My understanding, you can correct me if I'm wrong, but my understanding is they've set up the software where if you do prove out an animal as being a het pied, then it would go up. Like if that dad was a Pos het pied, it'll go up in your software and mark that as being a het pied. It'll just automatically take care of that for you.
Yeah, it does. It does update. Even we're getting the shed test results back on the animal that you've tested. It updates it straight away and then traces the lineage back. So it is. Yeah. It's a fantastic bit of software, and I think it's perfectly integrated to what you guys are doing.
Yeah, very cool. That's awesome. I know they've put a lot of time and effort and thought into that. It's an awesome product for ball python keepers.
And I think it's just scratching the surface at the moment. There's still so much more to do with it.
Yeah.
Hi there, this is Scott from Scott Wilkinson Reptiles. I'm pleased to support that Wolfie Royals podcast. It's absolutely fantastic to have a UK-based podcast, and Tom's slowly starting to get the hang of this. So, for those that don't know me, I've been breeding reptiles now for over 35 years and have focused on breeding ball python morphs for the last 20 years. I'm also working with probably the largest collection of Australian blue-tongue skinks in Europe, including albinos and melanistics. I also have been importing and exporting reptiles for over 25 years now and currently offer that service for other breeders and collectors, and can ship your animals in and out of Europe, USA and Canada. Best place to find me is on Instagram or YouTube, or if you're really unlucky, you'll see me half naked at a reptile show somewhere around the world. Message me via Instagram for any further details, and enjoy this amazing podcast. Take care.
So obviously there's pros and cons to the shed test, and then one thing we can't not mention is the desert ghost, scandal as such, if you want to call it that. So let's get into that and talk about where the findings come from, what the findings showed, and then where we are now.
Yeah. So I mean, we detailed it in videos, maybe in too much detail. And so I think people in general just kind of gloss over things, or they see somebody says something and they just, repeat that on or whatever. But if you go through and watch the videos, it's all laid out very, very detailed. So we found a hit that looked pretty good. It was right about, 80 to 90% of the time. And and people, you have to keep in mind, literally every single day, people are asking us, because we had, pied and clown and albino and ultramel. We had all this stuff come on pretty quick at the beginning. And we're like, okay, we're working on desert ghost. We're going to have desert ghost soon. And, it worked so well with clown. And then we have these others from the academic researchers that, seem to not be too difficult to find. And so we just, every single day, we're letting people know, hey, we don't have it yet. We're working on it, but we've got something good. And so we started letting people know, hey, this looks pretty good. It's right most of the time, but not all the time. And it seems like when it is wrong, it's just, one way or the other. So like if it says that it's a het, then most likely it's right. Or if it says that it's negative, I think that's what it was. If it said it was negative, that's most likely correct. If it says it's positive, sometimes that positive will be wrong. And so that's why we called it an elimination test, both on Morph Market and on Clutch. Because when we were telling people, hey, it's good most of the time, and then we found another marker that was on a different chromosome, and it definitely increased the calls as far as being correct. If we looked at what we were calling DGA and DGB, but they're on different chromosomes, which would make it a polygenic trait. But when we looked at those two markers together, that helped us increase the accuracy of the call. And so it was more like 85 to 90% correct. And so we're still telling people, hey, it's better with these two markers. It looks like there's more than one thing affecting desert, what makes this a desert ghost, like it's a polygenic trait. And then when we use these two pieces of information from completely different chromosomes, that increases our chances of getting the correct call. But it's still only like 85, 90% correct. And people are like, well, I don't care. That's way better than 50% posset or 66% posset. At least then I would know I'm more like 85 to 90%, sure. And so eventually we're like, okay, as long as we put it on Morph Market and Clutch as an elimination test and let people know, hey, that's our best way to show people when they go to buy it, it looks different from all the others because it says elimination test. So this is not as accurate as the others. So if you buy this and you get a result, just know it's not over 99% correct like everything else. And then we also on our show said, hey, any of you that have animals that at least one of the parents was a desert ghost, one or both. So it should either be a visual desert ghost or a het desert ghost. Send that in because we know that we don't have the whole story. Before we knew what DGC was, we said there has to be a DGC. We don't know where it is. So we're still looking for it. So send us in. So we ran 2 different DG tests, ended up being 3 different DG tests for over 500 samples for free. And that's, a lot of time and money. But that's another thing. We're showing people like, hey, if you think about this a little bit, they're calling it elimination test, letting us know it's not as accurate as everything else. And they're doing a whole bunch of tests for free. Like we're trying to be as transparent and clear as possible. We're still figuring this out. Yeah. People want something that they can purchase and help them make breeding decisions, but we're not there. And so when we got all that information, and then we got another round of sequencing back, and we did find another marker, what we call DGC, and it ended up that DGC by itself calls it. So we don't have to look at A and B anymore. DGC is it. So it really is just a single mutation like clown and pied and other things. That DGC was the one. So what we had incorrect was we thought that since we picked up A and B first, they probably affected what makes something DGC or makes something a visual DG. That probably had the biggest effect since we found them first, but it ends up DGC that we found third actually had the biggest effect. And so once we had that, we let people know, and we have, YouTube videos all about it. Like, this is DGC. It's the one. People in Canada and Europe saw the same thing where it's just one mutation for both DG and the other
enhancer.
Yeah, enhancer. Same thing. It tests the same. It's one gene. We were calling it DGC because it's the third one we found, but we were very, very open and detailed with the information the whole way, and all we were trying to do is figure out, what's going on. And what we learned from that was, no matter how much we try to keep people informed on what we're doing and why we're doing it, and when we figure out exactly what's going on, when somebody goes on that, maybe doesn't watch YouTube or doesn't do this or that or whatever, they go on and pay for a genetic test, they might not realize that elimination thing there, that isn't on all the others. And, anyway, people got very frustrated. So we learned, okay, we just won't let people be a part of that journey. That was clearly a bad idea. I'm thinking about it from my standpoint, being in genetics and breeding for over 20 years. And that was how I would have liked to have been treated. But I can definitely see now, from other people's standpoint that, if I go on and I pay for this genetic test and this is the result, and now I'm getting these results that say A and B, and then later there's the C, and, I can definitely see where it was confusing and frustrating, and certainly was not our intention. And if we had known it would end up like that, clearly we wouldn't have done it that way. But what we were trying to do, since we had people contacting us, literally multiple people every single day, and saying, I don't care if it's 85%, I want to be able to pay for it and use it. We're like, okay, we'll do it. And we'll make sure we, are always letting people know what's going on, but we won't ever do that again.
Do we have any idea what was causing those, like the hit markers on DGA and DGB? Was there something that is causing those, or determining you to look there as that marker?
So with DGB, that's on a separate chromosome, and so that might have something to do with color or pattern. From what probably the person I've run the most for is Justin, and he said that he doesn't really see anything on ones that were like homozygous or heterozygous DGB, but were desert ghosts. He doesn't see anything. Like, I thought maybe it could be a contrast or a color difference. They're not seeing it. It could be that it just helps the animal be a little more healthy, and so it stayed in that, somehow that got selected for, at least in the United States, desert ghosts. It wasn't something that they really saw in the ones in Europe and Canada so much. So with DGA, the answer there is DGA and DGC are on the same chromosome. And so DGA was a marker that had a high correlation, was right most of the time, but it was not the actual one that causes desert ghost. And so what happens, if you remember from high school biology with crossing over, every once in a while, it's like banana and sex. 9 out of 10, you're going to get a male maker banana. The bananas from that are going to be males, but 1 out of 10, it will be a female. That's kind of the same thing with DGA and DGC. Most of the time, if you get one, you're going to get the other, but every once in a while, there will be a crossover event and those will get split up. And then that DGA marker is no longer a good marker for actual desert ghost. And so that makes sense. DGB, we don't know if it gives some kind of beneficial either look or health, or why there was such a high amount of the desert ghost animals in the United States that had that. Don't know. But it definitely seems clear. DGC, and it's the same one that they're testing for in Europe and Canada. It seems like it's the one and works really well. And we've run it in lots and lots of panels, and it seems to match up with ones where people already knew it was a DG or a head or whatever. It's done very well for us.
Were there any other genes that you tested for that had those sort of hit markers on different areas that were maybe a little bit misleading?
Not like DGA, DGA/C. So that was on one chromosome, and then DGB. So most of the time, when we get the readout back from the computer, it has all the chromosomes lined up, and we can look across and be like, okay, in chromosome 7, we see this big peak, and that's the only place we see the big peak. Okay, that's the chromosome that mutation's on. And all the others we had worked on, we just go through, oh, there's one big peak. That's the chromosome we need to focus on. With DG, we had 2 peaks, and that was really weird. And I don't I have not seen that since, where we have that, really nice double peak going on. So it just is something weird.
I think it just happened to be that Desert Ghost was the in gene. It was the hot gene at the time that everyone was waiting on, and everyone was breeding and waiting for results. That if it had happened on a gene like albino, no one would really been that bothered by it. No one would really known the difference, and they would probably just continue to breed as normal and almost like rubbish the data that was coming out saying it was polygenic. But I think where it was Desert Ghost and it was so in demand, it was like this huge issue that was going on.
Yeah, but I mean, we answered people's questions whenever we could, wherever we could, and definitely put out in the YouTube videos, the Reptile Genetics Weekly YouTube videos, all the information. Everything was as clear and transparent as we could possibly make it. But there's just, some people that don't look at that stuff. And like I said, I can understand from their point of view. If I pay for a genetic test, I expect it, to be clean, simple, easy, and correct. And, that was not what we were offering customers. And now we know, no matter how much we talk about it, there's going to still be a significant percentage of the customer base that is not going to see that information.
Yeah, that's true. How did that affect business? Was there a downturn in business at that point, or is it remained pretty steady?
No, it's steadily grown the whole time. And like I said, I mean, we answer people all the time and, let people know. And once we knew about DGC, we went back and ran, people's samples where we had run DGA and DGB. And still to this day, if you have one, if anyone listening, if you have one, you only have DGA and B results for, all you have to do is send the shed in again and let us know you only have A and B for it, and we'll run C for free, So I mean, it hasn't slowed things down. No, plus there's so many other mutations that people are interested in. And so, it's not been a problem. People, the people that were the most concerned did, access the information, understood what happened. And, since there's competition, there'll be people that will, try to say, well, that's why maybe this other company's better or whatever. But I mean, if you're the first to do it, the first to try it, and sometimes these things happen, there's cell phones that blow up and trucks that start on fire, go on fire. There's, recalls, there's things, and all kinds of companies that have been, successful for decades and continue to be successful. There's stuff that happens. The biggest thing we learned is, we just can't have a test be available. No matter how much we talk about it being different from the others, we just can't have a test be available to customers publicly unless it's over 99% accurate. Yeah. It's just not something that people can handle.
Andrea Lupodini is one of the leading names for graphic designs and logos within the reptile industry. He produces high quality designs for some of the most recognizable names and faces within the hobby and the industry, and he has also designed my logos too. Branding and marketing are so important these days, and making sure you stand out from the madding crowd is difficult, or it was up until now. If you're looking for a new logo, or perhaps you just want to revamp your existing design, then look no further. Andrea will work closely with you to bring your ideas to life, and he can do so in a variety of different styles also. As well as logos, Andrea also designs eye-catching roller banners, social media banners, and YouTube banners, to name but a few, and these will all give your pages a fantastic professional feel and edge. For a full range of products and services available, check out Andrea Lupodini Design on Instagram and Facebook, but please be sure to look out for that blue verification tick. This series, we're excited to bring you a unique podcast bundle to really up your game. Purchase a full logo design and receive 2 additional services free of charge. This could be a Facebook cover design, a table banner, a roller banner, stickers, t-shirts. There's nearly 20 extra services and products to choose from, so you can tailor the package to suit your exact needs. And the greatest thing about it is you'll even save yourself over €100 too. So, use the promo code WOLFIE24 when inquiring with Andrea to activate your podcast pack savings today. Be sure to follow Andrea's social media pages for further fantastic offers, and check out all of his existing deals too. Thanks for listening. Now back to the podcast.
Hi, this is Darren from Wild Rex. We've decided to sponsor That Wolfie Royals podcast for the second year running, as he done so well last year. So if you're in the market for a custom made rack in any colour you want, with any colour tubs you want, check us out on Instagram and Facebook. Back to the podcast.
Have you ever had a test come back that say has come back from a visual pied that's come back negative?
Yes. And so the very first time that happened, I think it was in the first 1 or 2 runs. And I asked the person, okay, so what's the information on this animal? And she's like, okay, it's a female pied, and she was running it for pied and clown, I think was what it was. And it came back as being negative for pied and homozygous for clown. And she sent me the picture. It was clearly a pied. And I was like, yeah, I don't know. And then I said, okay, so you said this is a female pied. So I ran a sex test and saw that it was a male.
Oh, wow. Okay.
So I already knew, okay, there's a problem here. But yeah, it ended up that the sheds just got mixed up. She sent me a male clown shed when she thought it was a female pied.
Yeah, that's not ideal.
So I have not had so when we're talking about thousands now, okay, so thousands of samples. We've had a single clown test come, and the animal's clearly a clown. And this same thing happened with a single genetic stripe female, and then her offspring didn't test like they should. So I'm talking like a single animal for clown, a single animal for genetic stripe that did not test correctly. And so we had them, send sheds again, make sure we're running the right shed. And, that's multiple times. So I'm right now, actually, literally this morning, I picked up a new clown test where I've just moved the primers a little bit. Because what can happen is there could be a mutation. So like I was talking about earlier, if the mutation's right here and we design primers on either side, that mutation is what we care about, but we have to have the primers on each side so that we know we're essentially saying this is the chunk of DNA we need to look at. But if there happens to be a random mutation pop up where one of those primers are, then it won't work anymore.
It doesn't go far enough.
Exactly. It can mess that up. So if one copy in that animal is like everything else, but the other one has a mutation where that primer is, then I'm only going to get amplification from the one that says it's het for clown. But since the other one that would have told me it's normal at the other location doesn't amplify, then it looks like it's a clown when really it's a het. But like I said, that's one out of like tens of thousands. And so I shifted it a little bit. And so I'm going to have to try it on a whole bunch and make sure that there isn't another line someone has somewhere else that has a primer or has a mutation somewhere. So it's extremely rare. For everyone else, the clown test and the genetic stripe tests, have worked well on all the panels and all that stuff. So it's extremely rare. We're talking like I said, 1 in tens of thousands.
Yeah. So one question I do get asked a fair bit with sending the sheds over to you guys through Clutch is the best way to prepare and store a shed for testing. So you hear a lot of theories about like sort of air drying it, naturally drying it, freezing it, etc. What is the best way to keep that DNA source stable?
So the number one thing is to not let it be wet or moist for very long. So when I'm looking for sheds, I check every morning and night. So it's never more than 12 hours that it's possibly sitting there. And even when I do, if I go and part of the shed's all crumpled up, but there's a part that's, stretched out, I'm going to go to the stretched out part and just tear a little piece out, like about the size of a quarter. That's the most clean and dry when I find it. And then I'm going to set it somewhere, let it completely dry. I would rather you set it at room temperature and let it completely dry than just immediately take it to the freezer. Having it be cold doesn't help keep the DNA stable. Having it dry is what helps keep the DNA stable. So I have sheds that I remove from the cage, fairly soon after they were shed, let them completely dry at room temperature. I put them in a Ziploc bag, and they've been sitting in a kind of moist basement in a Ziploc bag for, I think the last time I ran them, it had been between 6 and 7 years. I just open that bag up, get a little piece out, run it, and it still works. And that's at, room temperature. Since it's in the basement, maybe it's a little below room temperature for part of the year or whatever, but So yeah, it's really just the moisture, humidity, wet. Like if you go in and the whole shed is in the water bowl and you haven't been in there for 6 or 8 or 10 hours, that shed's crap. It's not going to work. That's the problem. So there's a lot of people that think that, heat or, whatever else, really none of that stuff. The DNA is pretty stable except for when there's water. The water will tear it up.
That's really interesting. So how does it work in, say, like more humid enclosures? So people are running cocoa, etc. Say it's not in the water bowl, but it's got quite a lot of excessive moisture from underneath and humidity, etc. Does that help degrade it down as well?
It definitely will. So like if you're gone for the weekend and it's sitting there in that, high humidity environment, it could be by the time Monday comes and you go in and pull it and you send it to us, it could be that, it already got chewed up over the weekend. One trick, like if it's a shed, you really, really want to make sure you get what you can do, watch them go into blue. And then when they clear up, so within the next couple of days, they're going to shed. Dump their water bowl out and just put barely enough to cover the bottom of the water bowl, or even just don't even have water in there for those 2 days. It's not going to hurt the animal if it's had access to water all the other days and you're going to put water right back in. But that could be a way you can make sure. But now that we've put out a video, shows where you can do scale clips. So if it's like a juvenile to an adult size ball python, it's really not too difficult to get just the very outer edge of one of the belly scales. It's clear and you just shave off. It literally looks like an eyelash. Just shave off the very edge. And you can go to our YouTube, look for the scale clip video. It's like 1 minute long. You can see how to do that. But if it's a juvie to an adult, you can just do 2 or 3 scale clips, put that in a Ziploc bag and send it to us. And those work really well. And then if it's a baby and the whole shed is in a water bowl and you don't want to wait, it's a bummer that it's all crap. The good thing about it, if it's that small, it's too small to scale clip, but it's also going to go into shed a lot faster than, an adult female that might be 4 months before she sheds again,
How important is it to keep the hatchling separate during the initial shedding process? Because I remember, I think when it first started with the shed testing, people said not to use the first shed, the first shed they cut. Is that still the case, or is there good enough DNA in there to test from that?
Yeah, I don't know for sure where that came from. It definitely wasn't us. You 100% can use the first shed. What I would do is when you see them go into blue, then if you didn't already, like if you separate them, right out of the egg, that's fine. But if they are together and they go into blue, the first 2 or 3 go into blue, that's when I would separate them. Because what we've found with garter snakes, where people keep them pretty wet, and there tends to be more keepers that keep garter snakes in groups.
Yeah.
When we do sex determination tests on garter snakes where they've been kept in groups, we've been able to prove that if there's a male and a female in there, sometimes that will cross-contaminate and we'll get the wrong result. So if you take them out, separate them when they go into blue, then you're usually 3 or 4 days out from when they'll actually shed. And that should remove any chance of cross-contamination. The shed itself is good to go. If you're leaving them in the incubator at really high humidity and they all shed together and you don't pull them out for a few days, obviously you don't know which shed goes to which baby, and the DNA has already been chewed up. So if you're doing that, that's definitely not going to work for you. You would have to change your procedures. But if you already remove them out of the egg, and then once they go into blue, separate them, then your first shed should be just fine. That should work well.
Okay, that's interesting. And is a scale clip better DNA than a test, the shed test?
Yes, as long as you give us 2 or 3. So sometimes I would say 1 out of, 15 or so will have a rerun. And if all you've sent us is 1 small scale clip, that might have gotten used for that 1 test. And unless we happen to catch that we need to rerun it before that gets thrown away, then, we could potentially run from that same one again. But if we didn't catch it in time, that might have gotten thrown away. So the only time a scale clip is a bad thing is if you only send us 1 small one that's so small we can't cut it in half. And usually if you give us a pretty good size scale clip, it can still look like literally like an eyelash. It'd be very, very small. A lot of the time we'll cut those in half and leave the other half in case we need it. But yeah, I would say if you're giving us 2 or 3 scale clips per animal, that is just as good, if not better, than a shed, because I don't know of any time where we've had a scale clip fail.
Okay.
Because the scale clip was degraded. It might be that there was something else wrong with the reaction that made it fail. And so we went back to that same scale clip again and it worked just fine. It wasn't the scale clip that was the problem. It was something else in the reaction. But we definitely do have times where the shed itself, no matter how many times, 3, 4, 5 times we try, because it's, an animal that's difficult for the person to get another shed or get it to us or whatever. And it just, no matter what we do, we can't get any good DNA out of it. So yeah, I would say if you have a juvenile to adult size one and you've got fairly steady hands, you can handle doing those scale clips. I would do scale clips above collecting sheds. But obviously if you walk in the room, they're in the middle of the shed and you can get a nice, really clean piece, let it dry immediately. I would not hesitate at all to use that. That should be just fine.
Well, there's no wait times for scale clips either. You don't have to wait for it to go into blue and to shed out, which with an adult could be a lot longer.
Yeah. So for me, when I'm proving out new tests and they're, mutations I have, I just walk into my snake room and I'll scale clip. Like just, a couple of weeks ago, I went out and scale clipped like 30 animals. And, it took me, I don't know, 15, 20 minutes. It's not very, I've done it a lot, so I'm pretty fast at it. But if you are doing multiple animals, what you want to do is just have a little rubbing alcohol, and you just, whatever, if you're using, some people will use fingernail clippers. I don't like doing that. I like using scissors like you'd use to pip eggs.
Yeah.
And so I'll get the scale clip from, the 2 or 3 from that one animal, get that into the Ziploc bag, seal that bag. Then I'll dip the scissors in the rubbing alcohol or any kind of alcohol you can get. That seems to be pretty easy and cheap. Put it in the rubbing alcohol, pass it over a flame, and just flame that and let that sit for 5 seconds just to cool down a little, 10 seconds, and then go to the next animal. That way you're not cross-contaminating between animals. That's the only thing you got to worry about if you're doing a bunch at a time.
Yeah, that makes sense. So what's the future look like for RGI? What's in the plans in terms of growth, and sort of where does the business go from here?
So like I said, in the next month or 2, we hope to be at about 60 tests or close to it for ball python morphs. We are looking to design morph tests in other species. We've already started to roll into that some. So it would be morph tests in other pythons and hog noses, boas, stuff like that. We also are working on more sex determination tests in pythons and some lizard species that do have sex determination from genetics, not the temperature dependent ones. And then we also are doing some whole genome tests where we're able to do just a low pass whole genome and learn a lot about that animal and how inbred it is, how closely related. Like if you do that with, maybe you have 7 breeders, do that with those 7 breeder animals, we'll be able to tell you which ones are the most closely related. So you can, like with dry mark on the indigo snakes, that's something really important to them, that they maintain as much heterozygosity or outbreeding as possible. So that will probably be the first whole genome test that will come out, will be a dry mark on one. But we plan to also have it for ball pythons. It's been difficult to get all the way there. We're getting fairly close, but in that case, you would be able to send one shed or scale clipping, set of scale clippings in one time. We'd extract that DNA and then we'd do a whole genome run on that. And as long as we're doing enough of them, we can do it pretty cheap. We just have to get the volume up, or we're doing a lot of those. If we're doing them for indigo snakes and ball pythons and green tree pythons, this and that, then we can get the prices down. But anyway, what you'd be able to do is just send that shed in or the set of scale clippings in one time. We do that sequencing and we have that data for that whole genome, and we would automatically, so say this is a year from now, we've got 60 ball python tests, you would get the sex, you'd get the call for all those 60 tests. But then like 2 years later, say we get, a brand new mutation, we get, I don't know, what's one of the the cool, really rare ones right now.
Let's say you got a monsoon test ready to go.
Yeah, let's say it takes us another year to get monsoon. So we finally get monsoon, and that animal is a possible monsoon. Well, instead of having to send that shed in and wait for the test again, you'd be able to just log in. What we would probably do is just have like a $10 fee or something like that. And you would just press, run again, and it's just going to run that data again for all the calls we have at that point. So you could just pay some amount and you would update it so that it has all of the genetic information. And so over time, a year from now, 5 years from now, you could choose when you do it. If you wait 7 years, there might be comprehensive information about, clutch size, expected clutch size and, average length and just all this stuff that we can eventually do once we've done lots and lots of this. Not only can we make the morph calls, but we could tell you a lot more about, how likely they are to have slugs or how likely they are to breed at 2 years versus 5 years. And there's a lot of cool stuff that they do in like cattle and chickens and stuff like that we'll be able to utilize once we've done a whole bunch of this whole genome stuff. So there's a lot of cool stuff coming in the future, and the sequencing costs, they're able to keep getting those to go down. So hopefully we'll get to a point where we'll have our software, our software will keep getting stronger, and then the sequencing costs keep getting lower. We'll hit a point where that'll be a useful tool for breeders and keepers.
Absolutely. I know you've got to sort of make a move pretty soon, so I won't hold you too much longer. But if people do want to contact you and have a look at what's going on and check out, all your updates and stuff, where can people find you?
The best places are YouTube, Instagram, and Facebook. So those three. If you like the more social media pictures and kind of short things, Instagram and Facebook are the good place to see us. If you're on YouTube a fair amount, YouTube's a good place. We have kind of longer, not any really long form content, but 10 to 20, sometimes half hour, 45-minute videos that will go into detail. But yeah, if you message us through Instagram or Facebook, we're pretty good at getting back to you. The email address that's probably the best is raregeninc@gmail.com. Sean does a good job of keeping up with that pretty much every day, unless he's sick or traveling or something like that. But yeah, the messaging through social media and that email address are probably the best ways to get to us.
Awesome. Well, I'm going to end it here. I'm going to ask you to hang around for a few seconds while I pinch the audio that's going to be downloading very, very shortly. I'm sure you've done plenty of these, so you know the score. But just like to say thank you very much for your time. Thanks for coming on and sharing some insights and some knowledge with us.
Yeah, thanks, Tom. I appreciate it. It's been fun.
No worries. Thanks very much, Ben. Thanks.
Hey guys, it's Mark from MJ's Pythons, one of the co-founders of The Ark. The Ark is now a proud sponsor of That Wolfie Royals podcast. The Ark, which stands for Amphibian and Reptile Club, is a multi-species gathering of reptile enthusiasts in the south of England. We come together multiple times a year to share our passions and have a great time together. The Ark is an event focused on diversity and community, just like the Wolfies podcast. If you'd like to join the Ark and take part in our in-person event, head on over to the website at www.amphibianandreptileclub.co.uk and become a member today. For any other information around the Ark, please do also check out our website where we'll have past events, upcoming events, and all the information relevant to the Ark and its events posted on the website. Thank you for listening to this short advertisement and listening to this amazing podcast, and we hope to see you all at the next show.